Bismillah ar-Rahman ar-Rahim. Thank you very much to the chairman as well as to the whole team of organizing committee of New Colloquium who have called me to deliver this important talk. We have, I think, 5-10 minutes extra. I'll take the advantage of it. As it has been nicely pointed out by Professor Sajid Makbul in the morning, that nowadays everything revolves around the gut. If you see a lot of upcoming medical literature of adult medicine, there's upcoming evidence that all chronic ailments, many of them are linked to gut health. And what in the gut is important is the microbes and the microbiome. This concept may be novel for some of us here, but as I've said, literature is full of all these research papers, and it's I would call it upcoming medical research. So what we are going to do in next 30 minutes, and I think I have a luxury of taking 10 minutes that Professor Fenton has kindly given to me. In next 30 to 40 minutes, what we're going to look at is what is human microbiome? Does the fetus actually harbors a microbiome, or the microbiome is produced after delivery? What is a healthy neonatal microbiome and what constitutes dysbiosis? What are the consequences of dysbiosis? And we'll just touch upon a few preventive strategies which may help a newborn to overcome this dysbiosis. As we look at the universe, and the vastness of universe makes us humble. Similarly, taking the same analogy, drawing the same analogy, we feel absolutely humane and humble by knowing the fact that most of our body is composed of microbiome. We have more cells of bacteria, fungi, viruses than our own cells. And the genes that the bacteria, fungi harbor are far more than our own genes. So perhaps I'm less of Talal myself, and I'm more of bacteria, fungi, and viruses on me. And that's a very important fact that has been highlighted since the start of this century. Science is just beginning to unravel many facts about microbiome, and it's rather hard to get our head around them. There are many aspects to the whole thing. When is microbiome developed? What environment plays its role in? But today I'm only going to confine myself to what it means for us as a neonatologist, and we are going to look into the perinatal aspects of this microbiome. Now, don't be judgmental. I'll just give you two sides of the picture, and we'll look into things, what evidence each school of thought has. So I'm disclaiming it here that we don't need to be judgmental, but there are two schools of thought. A newborn's microbiome, is it developed after delivery, or is it developed, does the fetus contains that microbiome even within the womb? This debate has been there for last, I think, 30 to 40 years now. And let's look at the two different views and two schools of thought. The first school of thought that we've been taught since our med school was that womb is completely sterile. The fetus lives in an absolutely sterile environment. And this age-old dictum had its roots way back in 19th century when research proved that fetus is residing in a completely sterile environment. And because of this belief was as a result of the fact that 90% of the amniotic fluid is completely sterile. However, I would just like to mention that all these studies were based on traditional culture techniques. All these studies were based on traditional culture techniques. Since the dawn of this century, people came up with another side of the coin, another school of thought, that no, the intrauterine environment may not be all that sterile that we think of. And there was a flurry of paper, and there were alternative hypotheses that no, the baby acquires all this microbiome within the uterus. What is the evidence to this new school of thought? The evidence was that many amniotic fluid sample contained bacterial DNA. This was from the amniotic fluid. Apart from that, the first pass meconium of the baby also had a similar sort of microorganisms in it. And the third place which had such organisms was also placenta. And placenta has its own niche of unique microbiome. It's important to understand that this new concept was based on cultures which were very, very precise and identified 16S RNA techniques. If we believe this hypothesis, where does actually this colonization and this microbiome comes from? There are three sources. First is the maternal gut, which contributes to this microbiome to a large extent. The second one is maternal oral flora, and that's the reason that even one of the etiology of preterm delivery is periodontal disease in the mother. And the third is maternal vaginal tract. So these are the three places from which the fetus may be getting its microbiome. Let's dive deeper and look into it. During the first and second trimester of pregnancy, the maternal gut microbiome changes and has abundance of Lactobacillus and Bifidobacterium, along with relative, I would use the word relative, increase in Proteobacteria. This microbiome then has its effect directly as well as through certain chemicals which are produced like small chain fatty acids on the fetus. And all this interplay, during all this interplay, there's a huge role of maternal environment, what sort of food she takes, what sort of lifestyle she's having, what are the cultural factors, and what sort of environment she is living into. So there's a huge interplay of things which is taking place while the fetus is developing in the intrauterine environment. Interestingly, till day before yesterday, I've given you one school of thought, but there is no consensus yet. This has just been published 3 days ago. According to this commentary, which has been generated by a whole lot of people from different parts of the world, there is still no consensus, and still we are in the corridors of uncertainty as far as the acquiring of neonatal microbiome is concerned. Because the counter argument is that these techniques, which are micro RNA techniques, they might be become only a low biomass of microbes. They lack proper techniques, and there is failure of evidence of bacterial viability. So I've given you two schools of thought. You don't need to be judgmental. The consensus is not out there, but that's how the microbiome starts shaping its place in the fetal as well as in the neonatal gut. Let's look at the evolution, how this microbiome has evolved over a period of time. Over a period of time, the microbiome has changed. The microbiome of our ancestors was very different from ours because they lived in a very different environment. They were close-knit communities. They used outside toilets. They shared utensils. Over a passage of time, we have also seen that our Westernized lifestyle, our habits, our food intake has shifted, and the microbiome is constantly changing. But what I am interested in, I am going to tell you in the next couple of minutes, is how does this interaction takes place perinatally and what consequences it has, and what are the factors that contribute to this microbiome. So what is a beneficial microbiome for a neonate? There are two organisms which, if the neonate harbors, may the neonate harbor it within the uterus or after delivery. One is Lactobacillus, Bifidobacterium. These are the good guys. If the neonate has them, very good. He'll have a healthy life and a healthy microbiome. On the other hand, if there is dysbiosis, then the bad guys predominate, and they are Proteobacteria, Enterobacteria, Clostridia, Streptococcus, Klebsiella, and we know a lot of them. Where are we moving with this evolution? Unfortunately, instead of having the right microbiome, the human race is moving in a different direction. Over last couple of years, we have seen that the stool pH has started increasing, and it has increased from 5.0 to 6.0, which means that there is now less of Bifidobacteria and more of pathogenic bacteria in it. And these sort of pathogenic bacteria are E. coli and Klebsiella, which we as neonatologists always fear and we are afraid of. And unfortunately, our newborns, we dread having Klebsiella in our NICUs and in our babies. So starting from the baseline, so if we have a good microbiome, we'll probably lead a healthy life. But if we have an altered microbiome, that's the baseline of dysbiosis, where dysbiosis starts settling and then has long-term consequences. So let's look at the factors which cause dysbiosis. We'll list them one by one. If a baby is born at home as compared to if the baby is born in hospital, the microbiome is very different. Those born at home have high number of Bifidobacteria and Lactobacillus. Those born in hospital have a sort of, I won't call it bad, but yes, a bit of compromised microbiome. I'm not suggesting deliveries should take place at home. Under no circumstances I'm suggesting it. I'm just telling you that what factors affect the neonatal microbiome. Mode of delivery, look at this. Bifidobacterium are the red ones. If the baby is born with normal vaginal delivery, there is good number of good bacteria. Unfortunately, if the babies are born with cesarean section, and we'll come at the end of my talk on cesarean section, what is the situation in Pakistan? I'll just give you a snapshot. But if the babies are born by cesarean section, look at the bad Proteobacteria in the microbiome. So, place of delivery, the mode of delivery, and our very famous jungle juice antibiotics, जो कि हम सुबह शाम हर जगह देते हैं, and we use it like peanuts. What effect it has? Those who had perinatal antibiotics, the Proteobacteria number was far high. Mind you, Proteobacteria are the bad guys, and the number of good guy bacteria were less. And what sort of antibiotics are very good friend and are very handy antibiotics जो कि हम सुबह शाम इस्तेमाल करते हैं? Cephalosporins and penicillins. Not even that, we've even seen that if babies have these perinatal antibiotics, there is predominance of antibiotic resistant gene. So by just prescribing perinatal antibiotics, we are damaging the microbiome. It has been classically seen that if we give ampicillin and gentamicin within 48 hours of birth, look at this yellow one is the Proteobacteria, the bad guy, and the green one is the good guy. Those who were treated with antibiotics like ampicillin and gentamicin within the first 48 hours of life had a high predominance of bad guys and a low predominance of good guys. On the other hand, the controls did not show dysbiosis. Taking it further, and I am sure following my talk, Dr. Huck is going to make you more wise on this. It's not only the selection and the start of antibiotics, it's the duration of antibiotics which also matters and damages the microbiome. It's an established fact that the more prolonged you give, and especially after 5 days, if you give antibiotics for more than 5 days, you actually increase the risk of necrotizing enterocolitis and dysbiosis. Since we are talking about drugs, another important medicine that yes, now people have, I won't call it stopped using it, but yes, there is an awareness, but just to create more awareness, look at this, if you use H2 receptor blockers, the moment you start H2 receptor blocker, the growth of Proteobacteria increases. Proteobacteria, bad guys. And the moment you stop them, the growth falls and the growth of good bacteria starts rising. So please, H2 receptor blockers are a big no when it comes to newborn health and neonatal care. We've seen in the morning, Surah Bakaraki, ayat number 233 was recited in the inaugural session, the role of mother's milk. What is written in Quran? I think there's no discussion even on that. And science is now proving with every coming day the new benefits of breast milk. If you start mother's milk, you end up with a healthy microbiome as compared to formula feed. If you start formula feed, the staph, E. coli, Clostridia starts to make gut their home and then progressively increases. Since we are talking about human milk, what in human milk actually promotes a healthy microbiome? It is the human milk oligosaccharides. Professor Huma is sitting here and she would know far more on it. that there are two sorts of mothers, one who are secretors who secrete a high number of human milk oligosaccharides. Human milk oligosaccharides are basically the prebiotics. They are not used and not digested by newborn's gut, but they act as a food for healthy bacteria. So this is basically a good lunch and a dinner for Bifidobacterium and Lactobacillus. And the greater those mothers who secreted high number of human milk oligosaccharides had a better microbiome as compared to those who secreted less amount of human milk oligosaccharides. A second thing in human milk is very important, which promotes a healthy microbiome, is Lactophren. It is bactericidal. It kills Streptococcus, Pseudomonas, and Candida. If it doesn't kill them, it hampers their growth by taking iron away, and the bacteria can't utilize iron. And it's a good bacteriostatic agent also. And there are some studies which say that lactoferrin-derived peptides was found to stimulate the growth of Bifidobacteria. Another important thing which we are more concerned about is prematurity. Prematurity per se is a risk factor for dysbiosis because the preterm gut is very different from a term gut. Look at the preterm gut. This is the preterm gut, and the above is the term gut. The term gut has got a nice mucus layer, good number of Paneth cells, while the preterm gut, the mucus is scanty. There are fewer Paneth cells, and these babies unfortunately already are in a compromised state where they've got indwelling catheters in them, you're giving them antibiotics. All these factors in prematurity puts the baby at risk of dysbiosis. Now that we've seen the causes of dysbiosis, I'll come on to consequences. I'm only going to discuss two important long-term consequences. I know Professor Khalid Haq is going to talk about NEC after my talk. So he's going to educate you on that aspect, but I'm going to only give you a bit of insight into what gut-brain axis is. As the newborn is developing, in the first 3 years of life, there are a lot of connections and neurons and synapses which are being made. And there's a direct connection of gut microbiome with this neuronal development process, and this connection can be in the form of humoral, immunologic, as well as direct vagal and neuroendocrine signaling. And if there is dysbiosis, it leads to long-term issues like autism, personality disorders, cognitive disorders, and even people think they're establishing links of Alzheimer's and Parkinson's with dysbiosis. And this is the critical window that you need in the first 3 to 4 years of life. Even before that, you need a healthy microbiome. What is the evidence to all what I'm saying? The evidence has come from mice studies. They've seen that if a germ-free mice is a mice which is being conceived in an environment which is absolutely germ-free. The mother is kept in a germ-free environment, and they've seen that in a germ-free environment, the mice had exaggerated hypothalamic pituitary axis, and if you replace this germ-free mice with normal microbiota, the axis is reversed and the changes are reversed. But this reversal only takes place if all this process is done within the first 6 weeks of life. We've even seen that if this germ-free mice is transplanted with human feces which are from an autistic person, the mice develop features of autism. And if you replace and if you rectify this microbiome, the changes are reversed. It's not only the mice, but we now have evidence from humans also. Those with autism and autism spectrum disorder, the microbiome was quite different. Look at this, the bacteroides and the firmicutes. Those who had autism had a bad microbiome as compared to those who didn't. There is also evidence that it affects cognitive development. In this study, the babies were divided into 3 clusters. The cluster 2 was the healthy cluster, which had bacteroides and bifidobacterium and the good guys. And we've seen that this cluster did very well on expressive language scoring, on receptive language scoring, as well as on journal cognition. Similarly, there is a lung-gut axis also, and a similar sort of microbiome is also present in the lungs. We have evidence from mice that if antibiotics are used which alter the gut microbiome, it's associated with increased airway responsiveness. If this is done in first 9 weeks of life in mice, and we've seen that those mice who had an altered gut microbiome had a high level of IgE, causing increased hyper responsiveness and asthma-like features. We have similar evidence from humans also, and we've seen there are a lot of literature and a lot of studies which have suggested that changes in microbiome diversity from birth to 3 months of age is associated with increased incidence of asthma in later life. For hardcore neonatology, people sitting out here, now with a bit of survival of newborns and upcoming neonatal units, we are seeing cases of bronchopulmonary dysplasia because babies are surviving. There's an association of altered microbiome with bronchopulmonary dysplasia also. Professor Fenton was mentioning about extrauterine growth retardation. Though the studies are weak, cause and effect relationship cannot be established, but who knows, there might be some link of gut microbiome with poor growth and growth restriction in later life also. So just last 2 slides before I wrap up. What is the prevention and what is the way forward? First of all, we need to realize that yes, there is existence of a human microbiome which plays its role. We need to take small steps like avoiding antibiotics. Lot has been said, but I think still lot needs to be, this issue needs to be addressed even at a larger scale also. 2 more important things which we can do is, we need to plan natural full-term delivery as much as possible. Natural full-term delivery. Unfortunately, the state in Pakistan is this, that in certain places, natural delivery is an accident. We go for cesarean section, our cesarean section rate is very high. It's one of the highest in the world. We need to control that. And coming back to Surah Bakaraki ayat 233, which was recited in the morning, if the baby has had a cesarean section, we can reverse the effects of dysbiosis by giving mother feeds and feeding the baby with mother feed and avoiding formula feeds. Because even if the baby has had a cesarean section, by starting mother feed from day 1, we've seen that you can actually reverse the effects of that little dysbiosis which has happened. So the last slide with my takeaway packet would be that one can choose wisely to have a healthy neonatal microbiome, there are two paths. One is that we make the baby move on this left side where there is dysbiosis. He ends up with problems, and who knows, for life. Alzheimer's, even cancers, irritable bowels, bowel cancer. There are things which are associated with a dysbiotic microbiome. And the other option that we have is we make him move on this path for him to have a healthy life. Thank you very much.